Welcome to Biology at Friyojit, my dear viewers. Today we will explore transposable genetic elements, or which is also known as jumping genes, and understand how they move within the genome to influence mutation and gene expression. We will discuss today bacterial transposons, ACDS elements in maize, P elements in fruit fly or Drosophila, and human retrotransposons like line, SINE and ALU sequence. These concepts are very crucial for undergraduate, postgraduate, CSIR-NET, GATE and other competitive exams, and they highlight the dynamic nature of the genome. Let's begin. So, what are transposable elements? Transposable elements are basically DNA sequences that can move from one location to another within the genome. They are also called as transposons or jumping genes. They are found in bacteria, fungi, protists, plants and animals, including human. They form a large part of the genomes, that is more than 40% of the human genome consists of transposable elements. Basically, they help in shaping the chromosome structure, regulate gene expression and create genetic variation. So what are the types of transposable element? Transposable elements are classified into three main categories based on how they move. First one is the cut and paste transposons. The mechanism is the element is cut out from one position and it is inserted into a new position in the genome. Movement is controlled or accompanied by an enzyme called transposase. The transposase enzyme is usually produced by the transposon itself. So the key features include there is no increase in copy number. The element physically moves from one place to another, and it may move to another location on the same chromosome and a different chromosome. Why they are called cut and paste? Because it works like a scissor and glue, cut from one place and paste into another place. And the second one is the replicative transposon or copy paste mechanism. The transposon DNA is replicated or copied and one copy remains at the original site. The new copy then inserts into a new site and it is also controlled by the enzyme transposase. Here the key features include results in an increase in copy number and there is a net gain of one copy. So both old and new copies stay in the genome. Why they are called replicative? Because the element copies itself before moving. So this is the image showing the replicative or copy paste transposon. Retrotransposons mechanism, the element is first transcribed into RNA. An enzyme called reverse transcriptase converts RNA back into DNA and the new DNA copy then inserts into a new chromosomal site. So the key features include movement involves RNA intermediate, it increases copy number and genetic information flow, that is from RNA to DNA, that is reverse direction. So why they are called as retrotransposition? Because it reverses the usual flow of genetic information. Normally we know that it goes from DNA to RNA, but in this case it moves from RNA to DNA. Types of transposons: retrovirus like elements, it is similar to retroviruses. It uses reverse transcriptase and it is related to viruses like HIV. Retroposons, they do not form virus like particles and they simply code, simply copy and inserts themselves. So cut and paste mechanism, replicative mechanism and retrotransposition. There are basically three types. How it moves: cut and paste, it physically moves and there is no increase in copy number and transposase enzyme is used here. Replicative transposition, it is copies and inserts into a new place and there is increases in copy number and the enzyme transposase is responsible for the process. In case of retrotransposon, one RNA intermediate is produced, it increases the copy number and another enzyme reverse transcriptase is used in this process. Transposons in bacteria. Basic idea: bacterial transposons are DNA element that can move within a chromosome and between chromosome and plasmid. Though first discovered in eukaryotes, they were first studied in detail in bacteria. What are the main types of bacterial transposons? We know there are three main types of bacterial transposon. First one is the insertion sequence or IS element. Second one is the composite transposons and third one is the TN3 like elements. So IS elements are small. There is no extra genes and they are simple in nature. Composite element is comparatively larger. It has no extra gene, but its complexity is moderate. And TN3 element is larger. It is complex, and there is also extra genes. This is the diagram showing the IS elements. So we have to know what happens, what are the key features of IS elements. IS elements are basically, IS means insertion sequence. They are the simplest type of bacterial transposons that can move within chromosomes and plasmids. They were first found in unstable lag gene mutations of E. coli, where extra DNA had inserted into a gene. When this inserted DNA was removed, the gene became normal again, showing that IS element cause mutations by insertion. IS elements are small, usually less than 2500 base pairs, and they carry only the genes needed for their movement. They have short inverted repeat sequences at their ends, which are important for transposition. IS elements produce an enzyme called transposase that cuts DNA and helps the element move to a new location. When they insert into DNA, they create short repeated sequences called target site duplications. Many copies of IS elements can be present in bacterial chromosomes and plasmids. IS elements can cause recombination between DNA molecules, such as integration of the A plasmid into the bacterial chromosome to form HFR strands. They also help spread antibiotic resistant genes, leading to multi-drug resistant bacteria. So now this is the image. This is a diagram showing the composite transposons. Then there are also involvement of IS element in composite transposons. So what happens? Composite transposons are formed when two IS elements insert close to each other in DNA. The DNA between the two IS elements can move together as one unit. In this way, the IS elements carry nearby genes along with them. So composite transposons are usually written with the symbol Tn. Example include Tn5, Tn9, Tn10, etc. In Tn9, there are two IS elements face the same direction, while in Tn5 and Tn10, they face opposite directions. The genes between the IS elements do not help in movement but are simply carried along. These internal genes often provide antibiotic resistance, which is important in medicine. Like IS elements, composite transposons form short target site duplications when they insert into DNA. In some cases, such as TN5, only one IS element makes a working transposon enzyme, while the other cannot because of small mutation. So this is TN3 element, the diagram showing the length of the TN3 element. TN3 element is a large bacterial transposon that does not have IS elements at its end. Instead, it has short inverted repeat sequences at both ends. When TN3 inserts into DNA, it forms short target site duplications like other transposons. TN3 contains three important genes like TNPA, TNPR, and BLA. TNPA gene produces transposase, which helps the transposon move. The TNPR gene produces resolvase, and the BLA gene gives resistance to the antibiotic ampicillin. TN3 moves by a replicative method, meaning it's make a copy of itself while moving. So first transposes join the donor and recipient plasmids to form a fused structure called a co-integrate, and TN3 is copied here. The co-integrate then contains two copies of TN3 in the same direction. Then resolvage acts at a special site, that is race site, and separates the cointegrate into two plasmids, each with one copy of TN3. So now we will we'll discuss about the transposons in eukaryotes. Eukaryotic transposons are DNA elements that can move from one location to another within the genome. They vary in size, in structure and behavior, and some of them are very common while others are rare. A few of them move by cut and paste mechanism, where the element is removed from one site and inserted into another. These transposons usually have inverted repeats at their ends. When they insert into DNA, they create a short target site duplications. Some of them produce an enzyme called transposase, which helps in their movement. So this is the differences between bacterial transposon and eukaryotic transposon. This is the diagram showing AC and DS elements in maize. AC and DS elements in maize were discovered by the American scientist Barbara McClintock, who noticed spotted and striped maize kernels. She studied changes in kernel color, especially in the alluron layer, to detect chromosomal breakage. But when a dominant inhibited gene CI was lost due to chromosomal breakage, purple spots appeared on the kernels, forming mosaic patterns. She found that chromosome breakage happened at a specific site controlled by a genetic element called DS or dissociation, and DS alone could not cause breakage. It needed another element called AC element or activated element, and McClintock suggested that DS could move from one place to another in the genome, causing genetic instability. Later, research proved that AC and DS are retrotransposons or jumping genes that can insert into the genes and cause mutations. So the AC element is about 4, 5, 60 base pairs long and produces an enzyme called transposase. DS elements usually do not have a working transposase gene, so they cannot move by themselves. The transposase made by AC helps DS move, and similar transposons such as HOBO also found in Drosophila. So this is the diagram showing the P elements in Drosophila. It's an important transposon. So P elements were discovered in 1977 when certain crosses in Drosophila produced abnormal offspring with mutations, chromosome damage and sterility. This condition is called hybrid dysgenesis. So there are two types of Drosophila strain. M strain do not have P element and P strain which have the P element. So hybrid dysgenesis happens only when a P strain male is crossed with M strain female. Other types of crosses give normal offspring. So the problem occur because P elements from the father become active in the eggs of the M strain female. Scientists found small DNA insertions in gene like the white gene, proving that P elements are transposon. P elements are basically present in many copies in P strain but are absent in M strain. A complete P element is about 2900 base pair long and contains a gene that produces an enzyme called transposases like other transposable elements. So complete P elements can move by themselves, but incomplete ones need help from a complete P element to move. P elements move only in germ cells, cells that form egg and sperm, not in body cells. P strain female produce special molecules like piRNA that control P element activity and prevent hybrid dysgenesis in the offspring. So this is the diagram showing the transposable elements in human like line, SINE, ALU elements, etc. So human transposons are mobile DNA sequences, also called as jumping genes, that make up a large part of the human genome and can copy and insert themselves into new locations in DNA. So line, that is long interspersed nuclear element. They are long transposable elements found in the human genome. The most important type is L1, which can copy and insert itself into new places in DNA. A complete L1 has genes that produce proteins needed for its movement. Humans have a few thousand active L1 elements, while many others are incomplete and inactive. Sometimes rare L1 insertions can cause genetic disease. SINE, that is short interspersed nuclear element. SINEs are short DNA element, usually less than 400 base pair long. They cannot move by themselves because they do not produce their own enzymes. Instead, they depend on line proteins to help them copy and insert into new DNA locations, and SINEs are very common in the human genome. ALU elements are the most common and active type of SINE in human. They are named after a restriction enzyme called ALU that recognizes specific sequence within them. There are more than a million ALU copies in the human genome, although most are harmless, and some insertion can affect genes and may lead to disease. So in conclusion, we can say that transposable genetic elements highlights the dynamic nature of the genome, from bacterial transposons causing mutations and antibiotic resistance to ACDS elements in maize and P elements in Drosophila, resulting gene expression, and line, SINE and ALU elements shaping the human genome. These mobile DNA sequences give detailed genetic variation and evolution. Understanding them is essential for UG, PG, CSIR-NET, GATE and other competitive examinations. Thank you.